Hi, this is Angela with Propel Cure for Crohn's disease. As part of our continuing series of interviews with scientists on the front lines of Crohn's disease research, we bring to you Dr. Benoit Chassaing. Dr. Chassaing is principal investigator with the Chassaing Lab, which is part of the French National Institute of Health and Medical Research in Paris. Dr. Chassaing's lab concentrates on trying to understand how environmental factors play a role in shaping microbiota, detrimental microbiota in our bodies, which can lead to intestinal inflammation. His lab has done many interesting studies. A couple of examples include the effect of diet on our microbiota, which is of course a big concern with many of us, as well as how the microbiota can potentially play a part in anxiety-related behavior. With that, we hope you enjoy our interview with Dr. Benoit Chassaing.
Okay, so thank you again for being here with us today. We really appreciate your time, and having spoken with you before, we know and we all think your research is incredibly interesting and important. And so I'm hoping just to start off, you could tell us a little bit about yourself and your research interest and why it was that you became interested in the topics that you're interested in.
Yeah, well, so first of all, yeah, thank you for the invitation. It's always really a great pleasure for me to discuss about what we are doing in the laboratory, and especially when it's talking to patients. So yeah, my name is Benoit Chassaing, and I'm a team leader in Paris, France. And my team is really having a very strong focus on the intestinal microbiota and the role played by the microbiota in chronic intestinal inflammation. And so yeah, before we go into a little bit more detail about what we are doing in the lab, so I did my PhD in France, and at the time I was working on one specific bacteria that's involved in Crohn's disease, which are a bacteria that's named adherent and invasive Escherichia coli. So it's a very bacteria with very strong ability to induce inflammation. And at the time, I was having very nice time working on this bacteria. But after, for my postdoc, so after I graduated from PhD, I decided to have a little bit more of I would say a broader overview of what's in our gut. And so this is why I decided to join a team in America led by Andrew Geverts, working on the intestinal microbiota. And so this it's been 10 years ago, and I'm still working on the microbiota now in my own laboratory. So the microbiota being the complex community of microorganisms, so viruses, fungi, bacteria, so all the living organisms that's in your intestine and that are playing a very important beneficial role, but can also drive chronic inflammation in some circumstances. And this is exactly what my team in Paris is trying to do, is to try to understand by which mechanisms the microbiota can turn to be detrimental for its host.
That's great. And this talk today with us is actually very timely, because I think just a few days ago you had a paper published in Gastroenterology, which I thought was incredibly interesting and I think relevant to a lot of patients and caregivers. And it's on the topic of dietary emulsifiers, which I know is something you've been researching for a while. So can you tell us a little bit about your work with emulsifiers, what they are, how they impact the microbiome, and what you found to date?
Yeah, so I mean, this whole story started close to 10 years ago. I mean, the initial publication on this topic was published in 2015. So yeah, it's quite a long journey. So everything started with, so when I joined Andrew Geverts' laboratory as a postdoc, we wanted to know by which mechanism, as I told you just before, by which mechanism the microbiota can drive chronic intestinal inflammation. So the microbiota is actually very good for health. It helps us to digest the food we are eating. It helps us to be protected against infection, against gastroenteritis. So it's very beneficial. But we know also in certain circumstances, and that's especially the case in Crohn's disease, the microbiota can actually drive chronic intestinal inflammation. And we wanted to understand why in some patients, the microbiota is becoming so detrimental that it can drive Crohn's disease and colitis. So we decided to focus on food additives, as you said, and one of the most common food additives that's used by the food industry are dietary emulsifiers. So dietary emulsifiers are mostly everywhere. I mean, everything that's packaged, everything that's processed or ultra processed is going to have dietary emulsifiers, because you need to have those emulsifiers to maintain homogeneity and extend shelf life of the product. So all the ice cream, chocolate bar, muffins, packaged bread, I mean, all those type of products are going to be loaded with emulsifiers in order for them to be put on the shelf. And so what we did initially was to work with mice and treat mice with those emulsifiers. And what we found was in mice that were genetically susceptible to develop colitis, which is the case also for Crohn's disease patients, you have a genetic aspect of the disease. In those mice, consuming those food additives were actually driving colitis. So we observed much more animal developing colitis when they were treated with emulsifiers. And not only there were more animals developing colitis, but also the colitis observed was much more severe in those animals compared to the untreated animals. So after we did a lot of follow-up study trying to understand a little bit the mechanism or how this is working, and we can discuss a little bit about that also. But just in a nutshell, what we observed was that really the microbiota is the key driver in driving and leading to the detrimental impact of emulsifiers. So basically what's happening is every time we eat dietary emulsifiers, the microbiota is going to recognize those food additives, and the microbiota is going to become a little bit more pro-inflammatory, a little bit more aggressive, and this is going to drive chronic intestinal inflammation and colitis in susceptible animals. So this was published, as I said, a couple of years ago in 2015. But of course, the next important step was to understand the human relevance of this, because everything was based on mouse, and we definitively wanted to have some human relevance to those findings. And so this is something that we did in collaboration with the University of Pennsylvania, so Dr. Gary Wu and Dr. Jim Lewis, which are very well known for their work on diet and IBD. And what we did was to take, so it's a low number of participants over a short period of time, but what we did was to treat participants either with an emulsifier-free diet, so a diet without any dietary emulsifier, or a diet that's supplemented with CMC, so carboxymethylcellulose, which is one of the major used dietary emulsifier in the country. And what we observed so this was done in healthy participants, because we wanted to start with healthy participants, so not people with Crohn's disease. And what we observed is in those participants, consuming CMC was sufficient to pretty dramatically alter microbiota composition. We observed also some impact on microbiota, I would say, aggressive and inflammatory potential. So it was a short-term study. We treated the participant only for 15 days. So of course, it was not leading to colitis or anything in those participants, but it's very encouraging to us. And what we are currently doing is doing a follow-up study on Crohn's disease population, because now we want to know if maybe by removing those food additives in the Crohn's disease population, maybe in some select individual, we don't think it's going to work for everybody, but very likely in select individual, stopping eating those food additives can actually be very good in preventing the disease. So yeah, this gastro publication is the first step, again, using, investigating it in healthy individual, but the next step, and we are currently working on it, will be to do that on the Crohn's population.
And that's incredibly interesting. I mean, in my own family, we haven't consumed an emulsifier over the last 5 years. So, it's something that I certainly believe in as a method of prevention and also to potentially not exacerbate existing IBD.
And there is other data that are pretty interesting. So there is other study focusing on diet in Crohn's disease. So diet in Crohn's disease is fascinating because it's becoming more and more popular. I mean, at the beginning, we are thinking that we are going to cure the disease only by using therapeutics and very costly drugs. And now more and more we realize that very likely in some select individual, again, I don't think this is going to work on everybody, but on select individual, if we can identify them, maybe having some targeted dietary advice is going to work. So there is a lot of, more and more work on diet and IBD, and there is some very interesting data using some specific diets that avoid emulsifiers, but also avoid other additives and other group of food, that it can be beneficial in Crohn's disease. So it's very encouraging, but we don't know yet if this is driven by the emulsifier or not. So this is why we are currently launching a new study focusing only on emulsifiers. So we will finally have the answer in a couple of years.
And I think that's excellent research. And I think a lot of people are interested in diet. You know, I'm a big believer in diet. We have used dietary therapy in our experience helpfully, even though we don't know everything. I think there is a lot to be said for what we do know about what a healthful diet, you know, lots of fruits and vegetables, unprocessed foods, very basic common sense things like that. That people can apply today. But I think as a mechanistic understanding, it's important to know what the role is of additives and of the microbiome. You know, one of the things that you hear talked about in IBD is sort of this concept of the many hits that it takes to develop IBD. So, you know, that you have to have, and I know you know this, but just for those listening, that you have to have a genetic susceptibility, but that's actually a small piece. And then beyond that, some environmental hits have to occur. And, you know, what are those environmental hits? And if there's a potential that the microbiome and potentially food and food additives play a role in that, I mean, I think that's an incredibly important thing for us all to understand, both for prevention and for treatment.
Yeah, I entirely agree. I entirely agree with those aspects, and this is exactly the case for dietary emulsifiers. And what we think is happening, and I mean we are still working on that, but this is again what our current mice-based research is showing is you need to have genetical predisposition to have colitis in mice model. You need to have some select microbiota to have the colitis, and you need to have other environmental factors such as food additives. And if you treat only with food additives, it's not going to drive colitis. And there is some animal model genetical predisposition, it's not sufficient, but it's definitely the combination of those factors that's going to drive colitis in mice model. And we think it's exactly the same in human, and hopefully we'll be able to have definite answer on those aspects very soon.
And so drilling down a little bit more, you know, we've talked about the emulsifier piece, but drilling down on the microbiome piece. So you mentioned your early research with adherent invasive E. coli. So there are a number of bacteria, and I think what's always interesting to us lay people who read some of this literature, there are different bacteria, and in some cases potentially even fungi, that have been identified as potentially contributing in some way to disease. What's your view on sort of individual bacteria, you know, F. prausnitzii or versus kind of the ecology of the microbiome, if you will?
I think all of those research are fascinating. I really don't think that Crohn's disease it's an infectious disease in a way that it's a single bacteria that can drive the colitis. But I think what's happening is, again, if you have some genetical predisposition, harboring some of those bacteria and eating processed food with additives, then you can have, you know, the combination of those factors that are going to drive colitis. And just to mention adherent and invasive E. coli, I think so there is a lot of study on this specific class of bacteria, and I think the average is between 30 and 30% of people with Crohn's disease is harboring this bacteria, and it's only 5 or 6% is in control in people without Crohn's disease. So what's telling us? It's telling us two things: is you don't necessarily need to have the bacteria to have Crohn's disease, because again, there is 70% of people with Crohn's disease that do not have this bacteria, so it's not sufficient. And also, it means that you can have the bacteria but not the disease, because there is 5 or 6% of the people having the disease but not having the bacteria but not the disease. So it means it's definitely is very likely playing a role in some patients, but it's definitely not sufficient to drive colitis in those patients. And what's very, very fascinating with this bacteria, and this is belonging to some follow-up study we did on dietary emulsifiers, we were very interested to understand why dietary emulsifier is not driving colitis in everybody in the same way. So we had some mice data for that, and we have also the human data that we just published yesterday in Gastro, demonstrating that really everybody is not responding the same, both mice and human, to emulsifiers. And so we wanted to understand a little bit more that. And what we found actually is that you need to have some specific bacteria in your microbiota to drive, to mediate the inflammatory potential of the emulsifiers. And adherent invasive E. coli bacteria is one of them. So basically, when we have mice that were completely protected against the detrimental impact and the inflammatory potential of emulsifiers, and then we just gave them, infected them with adherent invasive E. coli bacteria, then they were starting to develop inflammation. So it's really a very good example where you need to have this combination of bacteria, food additives, genetics to drive the colitis. And it's I think very, very fascinating, because what it suggests also is maybe we can identify and we can use microbiota data, and this is something we are more and more doing in the lab. We can use microbiota data to try to identify patients that are having a susceptible microbiota to emulsifiers and patients that are not. And maybe one day we'll be able to, by just looking at the microbiota, identify who should stop eating completely dietary emulsifiers and who can keep eating ice cream, and it's not going to impact their Crohn's disease anyway, because they are fully protected against emulsifier and their disease is caused by something else. So I think all those concepts are very, very fascinating. It also demonstrates how complex and multifactorial is the disease. But I think we need to keep our focus on those aspects, because at some point, I don't think we are going to have a magic landing and a magic bullet to stop and cure Crohn's disease at the same time in everybody. But I'm really, really optimistic, and I really think that at some point in the near future, we'll be able to identify subgroup of disease. That, okay, you should stop eat that. Okay, you should take this antibiotic. Okay, you should have a transplant. And I think exactly the same is going to apply for fecal transplant. I don't think it's going to work in everybody. I really, really think it can help to cure the disease in some select individuals. So more work is doing, and we are definitely working hard on those aspects.
So it's fascinating, and of course we look forward to the day that we have cures for some percentage and eventually for all. But you're right, it seems like, in speaking to researchers, that we're really looking at potential treatments or cures for 5 to 10% here, 10% there, and 20% here. So it is a multifactorial disease. So you mentioned pro-inflammatory bacteria. Do you conversely think there are certain bacteria that are beneficial and protective?
Yeah, definitely. So this is the case for Faecalibacterium postnitzi, for example. Sorry for the name, but it's a very well-known bacteria that's able to. It's associated with protection against recurrence in Crohn's disease patients, and we have some data also in the lab showing that maybe there is some member in the microbiota that can degrade and digest dietary emulsifiers. So you can think also maybe it's not. If I am eating emulsifiers and I'm developing Crohn's disease, it may be not because I am having pro-inflammatory bacteria. It's maybe because I'm losing bacteria that help me to be protected against those food additives. So yeah, I certainly think that it's going to be a matter of stratifying those patients based on what they are having in addition of other, or what they are lacking compared to other people. And for this, we are making pretty good progress in trying to understand all the different subgroup of Crohn's disease patients.
So one of the sort of challenges, if you will, that I often hear when it comes to microbiome research is that we don't know if it's a cause or a consequence of disease. And my lay response to that is that it's a false dichotomy, and it may very well be both.
Yeah.
What is your thought?
There is no doubt that it's both. There is now plenty of data showing that if you have inflammation, it's going to detrimentally impact your microbiota. And there is a lot of data showing that if you detrimentally impact the microbiota by dietary emulsifiers or by harboring some select bacteria, it can be sufficient to drive inflammation. So I really think this is this kind of vicious cycle where the one is going to feeding the other. And then once you are in this vicious cycle, Crohn's disease is going to establish and recurrence is going to appear and this kind of thing. So I really think it's going to be both, and we have to find ways of stopping this vicious cycle before it even starts, or being able to find a way of stopping this vicious cycle once it's established. But yeah, I really think it's a combination of both. Inflammation detrimentally impacting the microbiota and microbiota leading to intestinal inflammation. But for emulsifiers, for example, what we know for sure is the microbiota is a direct target of emulsifiers, and if you remove the microbiota, or if you transplant the microbiota from emulsifier treated to emulsifier non-treated, this is sufficient to drive inflammation. So we know that for at least for those type of food additives, we know that the microbiota is really, really central for the inflammation. But maybe also what's happening is, when you are when you're a teenager, you are eating junk food, and you are kind of hitting your microbiota and driving a pathogenic type of microbiota that's going to drive inflammation. But after you can stop eating those junk food and eat normally, you already have a detrimental microbiota that's going to drive inflammation, and inflammation is going to drive altered microbiota. And so even if you eat healthy at the time, maybe it's too late. So I think we have to definitely take both into consideration. But yeah, inflammation is altering microbiota, and microbiota is altering inflammation. But I think now it's pretty well accepted that it's a combination of both, and we have to understand how to stop this vicious cycle.
And in your research, have you come across or found anything, whether it's in mice or in humans, that would sort of be sufficient to stop that vicious cycle of inflammation?
Yeah, so what we found and again, going to the emulsifier story, is you definitely need to have the combination of food additives and select bacteria. So what we found is if we remove select bacteria from the microbiota, we can fully prevent from the detrimental impact of dietary emulsifier. So yeah, we think we are soon going to have to find ways of being able to make microbiota much more resistant to the detrimental impact of dietary emulsifiers. And another thing that we are currently working in the laboratory is, in your intestine, so there is the intestine, which is basically you, and then you have the microbiota on top of it, which are all the bacteria, viruses, and these kind of things. And between them, there is what we call a mucus layer. So it's kind of a gel that's here to kind of keep a no man's land on top of our intestine. And we know, and there is accumulating data, and it's not only from my lab, it's from multiple labs, but we know that the bacteria that interact at this specific mucus layer are very, very important. So we are currently working on some select bacteria that are able to fortify this mucus layer to drive a much stronger mucus layer, and this is sufficient to protect against colitis. So again, this is bringing the concept that maybe in some Crohn's disease patients, they are developing colitis because they have too many detrimental bacteria. And in some other patients, it's because they are lacking those beneficial bacteria. And yeah, we definitely need to find a way to use the microbiota to make much more stratification of the Crohn's disease patient. And this is something that we are highly working on, and there is other team working on that. Try to use the microbiota from cohorts, from Crohn's disease patients, to identify and stratify them in order to be able to give them the best advice to prevent and hopefully cure at some point the disease.
And I think that's incredibly important research. It's, I know, a difficult question, but we in our community are very impatient people. How far off realistically do you think we are from some of these potentially therapeutic applications?
Yeah, I don't know. That's a good question. I mean, sometimes, research is taking sometimes much longer than what we expect. But thinking about the emulsifier story, everything started in 2015, so 6 years ago, and now we are launching the first human trial in Crohn's disease patient to see the impact of emulsifiers in Crohn's disease patient. So it can also go fast in sometimes. So yeah, I don't know, but I really think that in the next 5 to 10 years, we will be able to use the microbiota to stratify the patient and have some microbiota-based dietary recommendation and therapeutic recommendation for their Crohn's disease. I'm really an optimistic guy, so I will say 5 years.
All right, I like optimism.
Yeah, I know.
And it's sort of it's tangentially related, but we know, of course, unfortunately, people with IBD have a higher rate of colorectal cancer as well. And it seems to be part of that story as well, correct?
Yeah.
The microbiome aspect. So
yeah, the microbiome aspect is definitely part of the story. So we know that there is the microbiota in select individual that having some species that can drive and nourish the cancer. We know that emulsifiers is also part of the story. So we had another study investigating the impact of emulsifiers consumption on colitis associated colorectal cancer. So the type of cancer that's associated with chronic inflammation, and dietary emulsifiers was also very bad. So it's not only promoting colitis, it's also promoting colitis associated cancer. And we know that this is definitely linked to the microbiota as well. So I think this is why the microbiota field of research is just fascinating, because not only we can modify it with diet or with probiotic, prebiotic, but also we can modify it in a way that we can impact a lot of disease. And we can, I mean, in the lab, we are highly interested in IBD and Crohn's disease, but sometimes we have projects also on the gut-brain axis, so anxiety, stress. We also have project on cancer. And I think this is why the microbiota field of research is really, really fascinating. And I know a lot of people joining the microbiota field of research, but I definitely don't know anybody leaving the microbiota field of research. So I think it's a fascinating field of research, and I think it's also opening a lot of possibility, because if you remind, when we published the first, when the first human genome was published, I think it was a little bit more than 20 years ago, we were thinking that maybe at some point we will be able to cure all the genetic diseases, but actually it's not that easy, because modifying the genome, that's not that easy. But modifying microbiota, you have a lot of way of modifying the microbiota. So when the field of microbiota is currently emerging, I think this is opening a lot of opportunities, because we can use fecal transplant, we can use dietary advice, we can use probiotic, prebiotic, again, focusing on select individuals. So I think this is a fascinating field of research, and I hope we can make great progress in the coming years.
So, and you mentioned another topic that a lot of patients and parents are interested in, and that is probiotics. Have you come across anything in your research in terms of probiotics? And there are a very limited number of strains and species that are commercially available today, but have you come across anything that you found helpful in your own research?
Yeah, so I think there is some, we are not currently having any probiotic project in the lab. We are having some prebiotic projects. So prebiotic, it's the type of food you are eating that can help to nourish the probiotic bacteria. But I'm sure probiotic, it's again eating the same aspects, that very likely in select individual, you can find some probiotic that will be highly, highly beneficial. For example, there is some probiotic that are well known to favor mucus production. So, you know, the no man's land layer I was referring to. So in patients that are having defect in this mucus layer, taking probiotic to fortify this mucus layer can actually be very, very big important actor in curing the disease in those patients. So we are not yet there. I mean, we are not yet at the point where we can find the magic bullet for everybody and the magic bacteria for everybody. But I know there is a lot of work going on probiotic, and Faecalibacterium prausnitzii is also one of them. But we think that if we can use this bacteria and engraft this bacteria in patients losing it, we can maybe prevent recurrence of the disease. So I know there is a lot of work going on in those aspects, and I think that's fascinating as well.
Yes, it is fascinating. And you mentioned probiotics and prebiotics, of course, which is, you know, fiber from things like fruits and vegetables, which I'm also a very big believer in that. But it does sort of suggest, going back to maybe there are groups of different patients, it does suggest that you have to have the bacteria that are going to, you know, utilize those prebiotics and generate anti-inflammatory compounds. And right now we don't know, you know, whether patients have those or not outside the research setting. Do you envision a time in the future where we will have commercially available microbiome tests? Because
I'm sure of that. I'm a true believer of that. So I know this is already happening, and I know you can have your microbiota analyzed by some company. I just really don't think we are at this stage yet. And if I analyze your microbiota today, I'm not going to be able to tell you much yet. But yeah, I mean, for sure, in the next few years, for sure, we will be able to identify which bacteria is missing in your microbiota that can help you to protect against colitis, which bacteria is missing. So this is why eating probiotic and eating fiber is not having any impact on you. Which bacteria are present that we should get rid of so you can stop worrying about dietary emulsifiers. So yeah, I'm a true believer of those aspects. I just don't think we are there yet, but yeah, I think it's coming, and we are developing some tools in the lab. So we are developing an in vitro microbiota system. So instead of always bothering people with clinical trial, we want to kind of reproduce their microbiota in an in vitro, so in the laboratory. So we just use a piece of their poop, and we are able to reproduce the microbiota in the lab. And yeah, this is exactly what this research is showing, is that something that's beneficial for me can be not at all beneficial for you, or even detrimental for you. And something that's detrimental for me can actually have no impact for you. And I think at some point we will definitely be able to use a microbiota, a little piece of your poop, to tell you what you should eat, when you should eat, and these kind of things. We are not yet there yet, but I'm sure we'll be there at some point. I'm again really optimistic guy.
That's good. There are some commercial companies in the US that claim to do some aspect of that. I do have healthy skepticism around these.
Yeah, it's really crude. I mean, it's really crude approach, but based on the bacteria you have, and everything is relative also. So what's maybe, I mean, it's not because you have a lot of bacteria of one type, but it's really good at the population level. So yeah, no, I'm also still pretty skeptical of this kind of aspect at some point. What I can tell you, for example, is if I have microbiota from Crohn's disease patient, from a cohort of 10, 20 Crohn's disease patient, I can definitively use their microbiota in our in vitro microbiota system to identify who is highly resistant to emulsifier and who is highly susceptible to emulsifier. But we are not yet at the point where you can just take a little piece, sequence, and stratify the patient. We have to do a lot of work to identify who is resistant and who is susceptible. And now the next step is to try to go to molecular markers. So instead of doing all the in vitro gut and culturing, which again, I don't want to go into too much techniques, but it's a lot of work. We want to be able to do some, this kind of very quick and easy sequencing testing where we can stratify patient based on this. And we are actively working on. So, I mean, at least for dietary emulsifiers, I think this is something that's coming in the coming years. Yeah, well, I mean, this is our hope.
And my last question, then I'll ask Annabelle if she has any questions to ask you as well. But my question is, I'm always eternally fascinated, and I joke about how as a parent, you know, I've had to learn a little bit about microbiology and genetics and nutrition and, you know, immunology and all of these things. It's clearly a big story with a lot of components. And the microbiome seems to be an important component of that, both as the pathogenesis of the disease and driver of inflammation. So how do you put the pieces together? I think is my question for you. You know, you're working on one important aspect, but there are all of these different aspects that ultimately have to come together to help an individual patient. And maybe this is the personalized medicine we all dream about. But how do we get to a point where we can begin to put all the pieces together for patients?
Yeah, so we are. Yeah, this is going to be a long way until we reach that. But I'm also having some kids at home, and basically I'm just trying to teach them. But I mean, everything that we are saying about eating healthy, eating very diverse, avoiding processed food, eating a lot of fruits and vegetables, and favoring fiber. I mean, food that are really high in fiber can also be very good. Avoiding using antibiotics if you can avoid to use antibiotics. So all these kind of aspects, even my 6-year-old son is pretty well aware of all of those aspects. So I think this is one piece of the equation, just opening our mind. And I mean, all of those aspects about the microbiota were completely not known at all when I was a kid, and when I was a kid it was so important to wash our hands and to clean everything and to remove all the bacteria. So I remember when I was a young kid, it was very, very good for the company to make products that are killing 99.9% of the bacteria, but actually that's very likely much more complicated than that, and there is a lot of good bacteria. So this is what I'm telling my son as well. There is a lot of good bacteria. So we don't really know yet how to put all of those things together, but we are working on it. And I think having awareness of how important in the microbiota and how important is what we are eating in impacting the microbiota, not only today, but it can have long-lasting consequences. I mean, this is not something we mentioned today, but what I'm eating today can have very important consequences later, and especially for young kids. I mean, what this is actually at this age, when you are, before 5, that everything you are eating is building your microbiota, is building your intestine immune system, and how you communicate with your microbiota. So it's actually a very critical period of time. So yeah, being aware of all of those aspects, I think it's really, really important for the future.
Yeah, and I just want to mention one thing. Sometimes when this topic comes up, people feel guilty or responsible. Nobody wants to be blamed, especially as a parent. And I personally never think about it in that way. I think about it in terms of, every new piece of information you get. As you said, when we were young, we didn't know many of these things. We're learning.
Exactly.
And I think it's just information to help us make better decisions. It's really not about, it's nobody's fault that they got IBD or Crohn's.
Exactly.
It's not about blaming anyone. It's really just about understanding what are potentially some drivers of this.
I entirely agree with that. And also I think, and especially in the case for microbiota and diet, I think what's really, really bad is all the excess. But having ice cream here and there in the summer, I mean, it's not going to drive Crohn's disease. But eating purely processed food for all your college time, maybe that's not the way to go. So yeah, we have to find the right balance also, and not having too much excess of those processed food which are very, very high in dietary emulsifiers. Yeah.
Agreed. Thank you. Annabelle.
This was a great discussion. Thank you so much, Dr. Chassaing, for spending the time with us to discuss your very interesting and instructive research. I couldn't help but think about the proverbial phrase, "You are what you eat," as quite appropriate and timely. Once again, thank you.